வணக்கம் மாணவ செல்வங்களே, மீண்டும் உங்களை முப்பரிமாணம் நிகழ்ச்சியில் சந்திப்பதில் பெருமகிழ்ச்சி அடைகிறேன். நம்ம இன்னைக்கு பார்க்க போற லெசன் பிரின்சிபல்ஸ் ஆஃப் இன்ஹெரிட்டன்ஸ் அண்ட் வேரியேஷன். நம்ம போன எபிசோட்ல பிரின்சிபல்ஸ் ஆஃப் இன்ஹெரிட்டன்ஸ் அண்ட் வேரியேஷன்ஸ் பாடத்தோட சில கான்செப்ட்ஸ் பார்த்தோம். இன்னைக்கு அதன் தொடர்ச்சியாக மேலும் சில கான்செப்ட்ஸை சந்திப்போம். ஃபர்ஸ்ட் கான்செப்ட் செக்ஸ் டிட்டர்மினேஷன், பால் நிர்ணயம். So what is sex determination? So it is the method of determining the sex of an individual. ஒரு தனிநபரின் பாலினத்தை ஆய்ந்தறியும் முறைக்கு பேரு sex determination. It is the method of determining the sex of an individual is what we call it as sex determination. நீங்க எல்லாரும் படிச்சிருப்பீங்க. So in our body we have 23 pairs of chromosomes. மனிதனோட உடம்புல ஒவ்வொரு செல்களிலும் 23 ஜோடி குரோமோசோம்கள் காணப்படுகின்றன. So out of these 23 pairs, the first 22 pairs are called as body chromosomes, whereas the last pair, that is the last 2 chromosomes, are called as sex chromosomes. Body chromosomes are also called as autosomes, whereas sex chromosomes are also called as allosomes. தமிழ்ல சொல்லும்பொழுது உடல் குரோமோசோம்கள், பால் குரோமோசோம்கள் அப்படின்னு நம்ம சொல்லுவோம். So these sex chromosomes is responsible for determining the sex of an individual. இந்த sex chromosome இரு வகைப்படும். One is homomorphic, another one is heteromorphic. Homomorphic, that means homo means similar or same. Morphic means external appearance, வெளிப்புற தோற்றம். So வெளிப்புற தோற்றத்தில் ஒரே மாதிரியான அமைப்புகள் கொண்ட ஒரு வகையான குரோமோசோம்க்கு we call it as homomorphic, whereas heteromorphic chromosomes will have different in structure, different structural adaptations. So the sex chromosomes are of two types. One is homomorphic, another one is heteromorphic. Homomorphic chromosomes will always produce homogametes, that is the gametes will be very similar in their nature, whereas heteromorphic chromosome produces different types of gametes. That is, they produce the gametes which differ in their structure. So this homomorphic and heteromorphic chromosomes, how it has been explained அப்படின்னு நம்ம பாடத்துல மிக தெளிவா நமக்கு கொடுக்கப்பட்டிருக்கு. என்னன்னு ஒன் பை ஒன்னா பார்ப்போம். So heterogametic sex determination. So முன்னமே பார்த்த மாதிரி heterogametic, hetero means different, gametic means இனச்செல்கள். So இரு வகையான இனச்செல்கள் தோற்றம். Heterogametic sex determination. In heterogametic sex determination, one sex produces similar gametes, whereas the another sex produces different types of gametes. Right? For example, மனிதர்களே எடுத்துக்கோங்களேன், human beings. If you see human male, he is a heterogametic male. He produces two different types of sperms. One sperm will carry X chromosome, another sperm will carry Y chromosome. Whereas in case of human females, she produces egg cells and all the egg cells are similar, that is they carry only the X chromosomes. So we call the human females as homogametic, whereas the human males are heterogametic. Now in this heterogametic sex determination, we are going to see the two different types. One is heterogametic males, another concept is heterogametic females. Heterogametic males, we have two further different types. One is XXXO type and the other one is XXXY type. Let us see in detail, right? So look at the chart, and this chart depicts the XXXO type of sex determination. One such example is cockroaches. So if you see the image, the male contains autosomes plus only one X chromosomes, whereas the other X chromosome is absent or Y chromosome is absent, that is they have only the X chromosome. It is not a paired X chromosome. Whereas if you see the female, they have autosomes and a pair of X chromosomes. So while gamete production, so you know during the gamete formation there occurs the meiosis, right? So as a result of meiotic division, the sperms produced are of two types in males. One is autosomes plus one X chromosome carrying sperm, whereas another sperm contains autosomes only, that is they don't have the chromosome, sex chromosome. Whereas in case of females, all the eggs produced contain the autosomes and one X chromosome. So this type of sex determination is what we call it as XX-XO type of sex determination. Another example for this heterogametic male is XX-XY type. So this XX-XY type is also called as ligaeus type of sex determination, right? So this is most common in human beings, and also it is noticed in Drosophila. So if you see the human sex determination, the females contain autosomes and 2 X chromosomes, whereas the males contain autosomes and X and Y chromosome. Here, because of the presence of 2 different sex chromosomes, one is X chromosome and Y chromosome, during meiotic division, the gametes produced will be having, the sperms will be having 1 X chromosome sperm and another sperm will be having 1 Y chromosome. Here also, the males are heterogamete, whereas the females are homogamete, and all the egg cells produced by the females have only the X chromosome. That is why we call this as a heterogametic male type. That is XX-XY type, which is also called as ligaeus type. So இந்த கேள்வியை நம்ம 3 மார்க் கேள்வியா நம்ம எதிர்பார்க்கலாம். Write a note on ligaeus type of sex determination in human beings. இது வரைக்கும் பார்த்த இந்த ரெண்டு, that is XX-XO type and XX-XY type, இந்த ரெண்டு வகையிலுமே males produce 2 different types of gametes, whereas அடுத்து பார்க்க போகக்கூடிய heterogametic females அந்த condition எப்படி இருக்குன்னு பார்ப்போம். In heterogametic condition females, the females will produce 2 different types of egg cells, whereas the sperms produced by the males will be of similar in structure. இதை நம்ம confusion ஏற்படக்கூடாது அப்படின்றதுக்காக, to understand in a better way, the X chromosomes are denoted by the letter Z, whereas the Y chromosomes are denoted by the letter W. So இது நம்மளோட convenientக்காக, understanding capacityக்காக they modified. ஏன்னா அங்கேயும் XX-XY போட்டு இங்கேயும் XX-Y போட்டா there may be a lot of confusion. To avoid the confusion, instead of using X, the alphabet Z is used, whereas instead of using Y, the alphabet W is used. So that is why this methodology is called as ZOZZ type of sex determination, which is common in butterflies and other insects. Right? So if you look at the chart, here the males produce similar type of gametes, that is autosomes and only the Z chromosomes. Whereas if you look at the females, they produce 2 different types of X cells. One X cell contains autosomes plus 1 Z chromosome, whereas the other X cell contains only the autosomes, because neither of the X chromosome or Z chromosome is absent. Here the females produce 2 different types of X cells, whereas if you see the next chart, this chart we can able to see ZZ type of sex determination, which is common among birds. Here the males produce similar type of sperms, that is autosomes plus Z chromosomes, whereas in the females 2 different types of X cells are produced. One X cell contains autosomes plus Z chromosome, whereas another X cell contains autosomes plus W chromosomes. So that is why we call it as a heterogametic females. The females produce 2 different types of egg cells. In previous thing, that is ZOZZ type also, the females produce 2 different egg cells. One is the egg cell carrying Z chromosome and the another egg cell carrying no chromosome. Whereas here also the female produces 2 different types of egg cells. One carries the Z chromosome and the other carries the W chromosome. So these are the 2 examples for heterogametic female condition. Right? So now we have discussed about in detail about the heterogametic condition sex determination, right? Now let us see how the sex is determined in human beings. In human beings, the sex determination is purely under the control of Y chromosome. In males, you know, we have 22 pairs of autosomes plus X and Y chromosomes, sex chromosomes, whereas in females we have 22 pairs of autosomes plus a pair of X chromosome, which is the sex chromosome. During the gamete formation, right, meiosis will occur. As a result, the sperms produce 22 X chromosome, 22 autosomes plus X chromosomes. So here the male produce 22 autosomes plus X chromosomes, and also at the same time another sperm will carry 22 autosomes plus Y chromosomes. So here 2 different types of sperms are produced. Whereas if you look at the picture, females will produce X cells which are similar in their nature, that is they have 22 autosomes plus X chromosomes only. So when a X chromosome carrying sperm fuses with the X cell, giving birth is going to be a female child. Whereas if the Y chromosome containing sperm cell fuses with the egg cell, then the child is going to be a male child. So that is why I already told Y chromosome determines the sex of the individual. So Y plays a very crucial role in determining the sex. So in the Y chromosome illana namakku maleness endra content namak concept nama pesa mudiyadu, right? So the recent studies have discovered various structural genes present in the Y chromosome that is responsible for the determination of maleness. Let us see how. So Y chromosome and maleness. Y chromosomes are seen only in the male body, right? So Y chromosomes, it is a single chromosome. The top and the lower portion of the Y chromosome is called as pseudo-autosomal region, and that is the region which undergoes recombination with its pair, that is X chromosome, whereas the remaining portion of the Y chromosome does not undergo recombination. That is why the non-recombinant region of Y chromosome, the majority of the portion of the Y chromosome does not undergo any sort of recombination with the X chromosome, whereas the top and the lower portion, that is pseudo-autosomal region, poly udal paga region, Y chromosome il ulla poly udal paga region பகுதி, அந்த பகுதியில் மட்டும்தான் நமக்கு ரீகாம்பினேஷன் நடக்கும். The remaining portion, மிச்ச பகுதியில நமக்கு எந்த வகையான ரீகாம்பினேஷனும் நடைபெறாது. So that top and the lower portion is called as par region or pseudo-autosomal region. In that place only recombination occurs. But the majority portion of the Y chromosome contains the non-recombinant region or the non-recombinant region of Y chromosome, NRY region. That NRY region contains a gene called as SRY gene. That is sex determining gene of Y chromosome, sex determining region of Y chromosome. That SRY gene produces a protein product called as TDF. TDF expands as testis determining factor. This testis determining factor is responsible for producing the testis in the embryo. So we have to understand in a clear cut way. So Y chromosome, the majority of the Y chromosome contains a non-recombinant region, that is NRY portion. That NRY portion contains a gene called as SRY gene, that is sex determining region of Y, that is SRY gene. The product of the SRY gene is testis determining factor, TDF. Testis determining factor is responsible for the development of testis in males. So that is how the Y chromosome is very, very crucial for determining the maleness in an individual. So it is hence cleared that Y chromosome determines the sex of the individual in human beings. Inactivation of X chromosome. Inactivation, if you see, in males they have one X chromosome, one Y chromosome, whereas in females they have two X chromosome. So that is one more X chromosome is additional in the female's body. How this number is balanced? In males they have only one X chromosome, in females they have two X chromosomes. How this balanced in the change in the number is balanced? So that balance is acquired by its inactivation of the X chromosome. Even though the females have two X chromosomes, only one X chromosome remains functional or active. The remaining one X chromosome gets inactivated. That inactivated chromosome is referred as bar body, right? And this bar body was first discovered by Barr and Bertram in the year 1949 in the neuron cells of cat. So அவங்க தான் முதல்ல கண்டுபிடிக்கிறாங்க. Mr. Barr and Mr. Bertram in the year 1940 பூனையோட நரம்பு செல்களில் காணப்படுகின்ற அந்த சிறு சிறு dark spots, they identified and called it as bar bodies. But those bar bodies are nothing but the inactivated X chromosomes. Why that it gets inactivated? To make the balance, to compensate the balance. So that is why it gets inactivated. So it is clearly understood, if there are more than one chromosome, X chromosome in the female's body, one X chromosome will remain functional, the other X chromosome will remain inactivated. That inactive X chromosome is converted into a heterochromatin, darkly stained heterochromatin structure, what is called as bar body. And this bar body concept was first proposed by a scientist called as Mary Lyon. So hence it is called as Lyon's hypothesis. And moreover, one thing you have to keep in mind, the number of bar bodies will always be one less than the actual number of chromosome. For example, female oda உடம்புல பாத்தீங்கன்னா 44 autosomes plus 2 X chromosomes. அப்ப 2 X chromosome இருந்தா கண்டிப்பாக அந்த ரெண்டுல ஒன்னு inactive ஆயிருக்கும், ஒன்னு மட்டும் தான் ஆக்டிவா இருக்கும். So there will be, if there is 2 X chromosome, then there will be one bar body. Whereas in males, if you see the males, they have 44 autosomes plus 1 X and Y chromosome. There is only 1 X chromosome. So there won't be any sort of bar body. So இதை வச்சு நமக்கு கேள்வி எல்லாம் கூட அமைக்க முடியும். எப்படின்னு பார்த்தீங்கன்னா, for example, Klinefelter syndrome பேஷண்ட்டுக்கு பார்த்தீங்கன்னா, they have 44 autosomes plus XXY chromosome. அப்போ அவங்களுக்கு 2 X chromosome plus ஒரு Y chromosome. அப்படி இருக்கும்போது calculate the bar body for this chromosomal makeup அப்படின்னு கொடுத்துருவாங்க, 44 plus XXY. ஸோ அப்படி இருக்கும்பொழுது 2 chromosome இருந்தது, that is 2 X chromosomes. If there are 2 X chromosomes, 1 X chromosome will be get inactivated. As a result, there will be a formation of bar body. So எங்கெங்கெல்லாம் நமக்கு 2 X chromosome வருதோ அப்பெல்லாம் அங்க ஒரு bar body இருக்குன்னு we have to assume, right? So this inactivation of X chromosome is done to compensate the balance of the sex chromosomes. So நீங்க படத்துல பாத்தீங்கன்னா Barr, Bertram and Mary Lyon. So இவங்க மூணு பேரோட photographs கொடுத்திருப்போம். Haplodiploidy condition. So this is a special type of sex determination which is seen in the Hymenopteron insects, particularly in the wasps, குளவிகள் அப்புறம் தேனீக்கள்ல பார்க்கக்கூடிய ஒரு முறை தான் இந்த haplodiploidy, இரட்டைமைய sex determination முறை. So what happens? So in this condition, the queen bee has a diploid condition and the eggs produced by the queen bee is haploid as usual, right? So the diploid queen bee through the meiotic division produces haploid egg cells. When it gets copulated with the male drones, right, the fertilized egg will develop into female worker bee or another queen bee, whereas the queen bee can also lay the unfertilized egg, and those unfertilized egg will develop into the male drones. So the males among the bee colony are developed without the act of fertilization. That is why கலவியிலா இனப்பெருக்கத்தின் மூலமாக மேல் drones நமக்கு develop ஆகுது. That is why parthenogenesis, we call it as, through the concept of parthenogenesis, the male drones are developed by the queen bees, whereas the females, whether it may be a worker bee or it may be a queen bee, the females are produced only by the fertilized egg, whereas the males are developed only by the unfertilized egg. So we can able to see both the condition, that is haploid condition is noticed in male, whereas diploid condition is noticed in females. That is why we call it as a haplodiploidy mechanism, and this mechanism helps to increase the success rate of this bee colony. That is why it is called as kin selection. So this mode of sex determination contributes to the queen's reproductive success, because the queen in the beehive secretes certain hormonal substances which reduces or which suppresses the ability or fertility of the other female bees. That is why if you notice ஒரு bee colonyல ஒரே ஒரு queen தான் நம்ம பார்க்க முடியும், மீதி எல்லா femalesமே worker bees தான் இருக்கும். ஏன்னு பார்த்தீங்கன்னா இந்த queen bee secretes some hormone like substances which suppresses the other worker bees fertility. So always there will be one queen bee. So the other workers will provide all the support necessary for the queen bee, leading to the success of the beehive. And this methodology is what we call it as kin selection. இது ஒரு முக்கியமான கேள்வியா 3 மார்க்ல நம்மளால கேட்க முடியும். So next concept is sex linked inheritance. So நம்ம ஆல்ரெடி இந்த பாடத்தோட ஆரம்பத்தில் பார்க்கும்பொழுதே நம்ம சொன்னோம் inheritance. Inheritance means ஒரு சந்ததியிலிருந்து அடுத்த சந்ததிக்கு characters transfer ஆகிற அந்த methodology. That is what we call it as heredity, right? So the transfer of characters from one generation to the next generation is what we call it as inheritance or heredity. Here the name was given as sex linked inheritance. That is, the characters which are present in the sex chromosomes are transmitted. So how can we define? The inheritance of a character controlled by the gene located on the sex chromosome. The characters controlled by the genes which are located on the sex chromosome, that is getting transferred. That is why we call it as a sex-linked inheritance. So, the genes that are located in the X chromosome are called as X-linked genes, whereas the genes that are located in the Y chromosome are called as Y-linked genes, which can also be called as Hollandric genes. 2 mark la kepanga, what are Hollandric genes? Very simple. Y chromosome il kaanapadum genegal. So, that and the geneகளை தான் நம்ம என்ன சொல்றோம், Y சார்ந்த geneகள், Y chromosome சார்ந்த geneகள், அப்படி இல்லனா Hollandric geneகள் அப்படினு சொல்றோம். So, the genes that are located on the Y chromosome are called as Y-linked genes or Hollandric genes. So, in the sex-linked inheritance is most common among males. ஏனா மேல பொறுத்த வரைக்கும் there is only one X chromosome and one Y chromosome. இது ரெண்டுத்துல எதுல முக்கியமா X chromosomeல பாதிப்பு ஏற்பட்டதுனா கண்டிப்பாக அந்த கேரக்டர் மேலுக்கு எக்ஸிபிட் ஆகும். Whereas in case of female they are homozygous, that is they have two X chromosomes. ஒன்றில் பாதிப்பு ஏற்பட்டாலும் மற்றொரு X chromosome மூலமாக அவங்க சமாளிச்சுப்பாங்க. So அதனால usually the females will be the carriers of sex linked inheritance, whereas the males will be the affected people of the sex linked inheritance. நம்ம sex linked inheritance க்கு சில எக்ஸாம்பிள்ஸ் நம்ம பாடப்புத்தகத்துல கொடுக்கப்பட்டிருக்கு, என்னென்னன்னு பாப்போம். So examples of X linked gene inheritance. X linked gene inheritance means the genes that are located only in the X chromosome, right? X chromosome is a type of sex chromosome. So மூணு எக்ஸாம்பிள்ஸ் கொடுத்திருக்காங்க, one by one பாப்போம். First is red color blindness, which is also called as daltonism. The second one is hemophilia, இரத்த கசிவு நோய். The third one is Duchenne's muscular dystrophy. இது மூணும் X சார்ந்த chromosomal inheritance. The next one is Y-linked inheritance, that is nothing but hypertrichosis, right? One by one நம்ம பாப்போம். Hemophilia, இரத்த கசிவு நோய். So hemophilia, which is also called as bleeder's disease. So one markல கேட்கலாம், which is called as bleeder's disease. Hemophilia or thalassemia. நம்ம thalassemia தவறா பதில் அளிக்க கூடாது. Hemophilia, இரத்த கசிவு நோய். It is also called as bleeder's disease. It is a X-linked recessive disease. X chromosome சார்ந்த ஒடுங்கிய நிலையில் பெறப்படக்கூடிய ஒரு நோய் தான் இந்த ஹீமோஃபிலியா, X-linked recessive disease. And it was first reported by John Cotto in the year 1803. And in this hemophilic condition, what happens? நமக்கு தெரியும், ஒரு சிறு கட்டு ஏற்பட்டாலோ நம்ம கையிலையோ உடம்புலையோ ஏதோ இடத்துல ஒரு சின்ன வெட்டுக்காயம் ஏற்பட்டாலோ ரத்தம் சிறிது நேரம் வரும், அதுக்கப்புறம் உடனே அது blood clot ஆயிடும். So how does this clotting mechanism occurs? Because of a protein called as thromboplastin, right? So அந்த blood coagulation, blood clotting க்கு மிக முக்கியமான ஒரு புரோட்டீன் தான் அந்த thromboplastin. இந்த thromboplastin புரதம் இந்த ஹீமோஃபிலியா பேஷண்டோட ரத்தத்துல காணப்பட மாட்டாது, அப்படி இல்லனா மிகவும் குறைந்த அளவு காணப்படும். So that condition leads to the failure of the blood clotting and leading to the condition called as hemophilia. The hemophiliac patient will lack the thromboplastin substance, leading to the failure of the clotting mechanism. And this hemophilia occurs in a crisscross pattern of inheritance. Crisscross, குறுக்கு மறுக்கு வழியில் ஒரு ஜெனரேஷன்ல இருந்து, அதாவது குறுக்கு மறுக்கு வழி என்றால் என்னன்னா, முதல்ல ஃபாதர் கிட்ட இருந்து பெண்ணுக்கு, தன்னுடைய பெண்களுக்கு டிரான்ஸ்ஃபர் ஆகும், பெண்கள் கிட்ட இருந்து அடுத்த ஜெனரேஷன் ஆண்களுக்கு, ரைட்? ஸோ அப்பா வழியிலிருந்து பெண்ணுக்கு, பெண்ணிலிருந்து பேரனுக்கு. ஸோ ஃபாதர் டு டாட்டர் அண்ட் தென் டு கிராண்ட்சன். ஸோ திஸ் பேட்டர்ன் இஸ் கால்ட் as crisscross inheritance. It is not a direct inheritance from father to son. It is from father to daughter, then from daughter to grandson. So this pattern is called as crisscross inheritance, and this hemophilia mostly affects the male and it is uncommon among the female. Color blindness, நிறக்குருடுன்னு தமிழ்ல சொல்லுவாங்க. Color blindness, and this is also an X-linked recessive disease. And here, you know, for the vision during the daytime, the cone cells are very, very important, right? This cone cells, which are in a dominant condition, they can able to produce the cone cells, and we can able to see the vision during the daytime and the colored vision. Whereas in its recessive condition, the production of cone cells will be a failure. There won't be any sort of cone cells or a decreased ability of the cone cells. So they can't able to distinguish the colors. And that condition is what we call it as color blindness or color vision deficiency. It is a recessive X-linked disease. So a colorblind person cannot able to distinguish between various colors, particularly 3 different colors. One is red, green and blue. So we can able to see in the chart ஸோ நார்மல் விஷனுக்கும் கலர்டு விஷனுக்கும் எப்படி நமக்கு வித்தியாசம் ஸோ அவர்களுக்கு க்ரீன் கலராக இருக்கக்கூடிய ஒரு பொருள் சுத்தமாக க்ரீன் கலர் மாதிரியே தெரியாது ஸோ இட் இஸ் இன் த ஷேட் ஆஃப் black. Whereas in red color blind person, the red colored structure will totally appear in the structure of green. So this is how there is a deficiency in the colored vision. That is what we call it as color blindness. So next is hypertrichosis. So and this hypertrichosis refers to the condition of excessive growth in the ear pin. காது மடல்கள் நீங்க பார்த்திருப்பீங்க பெரியவங்களுக்கெல்லாம் பாத்தீங்கன்னா காது மடல்ல மிக அதிகமான முடி வளர்ந்திருக்கும். So that condition is called as hypertrichosis and this is most common structure which is seen among the certain males. And this is an example for Y-linked inheritance. The genes for this hypertrichosis condition is located in the Y chromosome. That is why we call it as a Y-linked gene inheritance. Example is hypertrichosis. Only the males are affected because the males are direct inheritors of the Y genes from the father. So that is why this disease, that is hypertrichosis, is noticed only in the males and the females do not contain this gene because the gene is located in the Y chromosome. Next concept is karyotyping. What is karyotyping? So it is a technique through which all the chromosomes of a particular cell are isolated and arranged in a particular fashion and it is photographed or drawn. That arrangement or sequenced arrangement of the chromosomes of a cell is what we call it as karyotyping. Let us see the definition. Karyotyping is a technique through which a complete set of chromosomes are separated from a cell and the chromosomes are arranged in homologous pairs, and this technique is what we call it as karyotyping. Right? So if you look at the image, and that is an example for karyotype. So நீங்க பாத்தீங்கன்னா நம்ம உடம்புல இருக்க 46 குரோமோசோம்களையும் homologous pair base பண்ணி arrange பண்ணி வச்சிருக்காங்க. Right? So that is a karyotype. So this image shows a very easiest step of preparing the karyotype which was proposed by Geo and Levon in the year 1960. So this is a stepwise procedure which is done to prepare a karyotype using the human lymphocyte. The first step is collection of lymphocytes from the human blood. Next is culturing of the lymphocytes. The third one is the induction of the mitosis. So after the mitosis occurs, that particular stage, that is the metaphase stage. You know, metaphase stage is the one where we can able to clearly notice all the chromosomes. So at the metaphase stage, the entire division is stopped by applying the colchicine. Colchicine is a anti-mitotic substance, right? It arrests the cell division. So applying the colchicine, the mitosis is stopped at the metaphase and the metaphase chromosomes are photographed and the photographed are arranged in a proper sequence to prepare the karyotype. So நீங்க இமேஜ்ல பாத்தீங்கன்னா karyotype நம்ம human karyotype மிக தெளிவாக கொடுக்கப்பட்டுள்ளது. Applications of karyotyping. Karyotypingனால என்னன்னு நமக்கு பிரயோஜனம், right? So மிக முக்கியமா karyotyping is used to identify the gender of an organism and also it is used to detect the chromosomal anomalies, to identify the genetic disease and also to understand the evolutionary relationship between the two different species. These are the essential benefits of the karyotyping. 2 mark கேள்வியில் நமக்கு இதை கேட்கலாம். List out any 4 applications of karyotyping. So மாணவர்களே, இன்று நாம் we have discussed about what is karyotyping, applications of karyotyping, sex determination in human beings, inactivation of X chromosomes, bar bodies, இந்த மாதிரியான கான்செப்ட்ஸ்லாம் நம்ம இன்னைக்கு பார்த்தோம். இதன் தொடர்ச்சியை நாம் அடுத்த எபிசோடில் காணலாம். நன்றி.